Hi, everyone. So I'm Marta. I'm working in protein production facility that it's in structural biology lab in Synchrotron. Okay. So last time, Andrea was telling you how actually the whole process of crystallization of solving the structures looks like. So I would like to tell you a little bit about how the process of actually getting the protein to do the functional and structural studies looks like. So maybe a little bit of introduction of myself. So I'm a biotechnologist. I did my master degree in Poland, in the University of Life Sciences in Bydgoszcz. Then I moved to Italy. I was working as a molecular biology assistant in the startup pharmaceutical company that was aiming on developing the diagnostical kits for cancer progression, and as well anti-cancer therapeutics. After this, I joined the laboratory of Elettra, of a group of Paola Storici, within the project that was called Proteo, that was aiming on creating the network of the collaboration between Italian and Slovenian laboratories. And we had let's say, the duty of providing the proteins for the partners that they were in need. And then within Elettra, I was following with the different contracts, let's say, but mostly I was as well responsible for production of the protein, setting up the high throughput facility, and as well setting up the platform for the insect cells production. And in the end, as well, as this is never too late to go back to school, I did my PhD later in my years, and it was in the biomolecular sciences in University of Trento in collaboration with Graziano Loli. And in the end, I came back to Elettra Synchrotron as a researcher and scientist, always in the field of protein. So yes, previously as well, Paola was describing a little bit protein facility, what it is, what kind of projects we are focusing on. I mean, we are really on a collaborative, let's say, way of thinking. So during these years when I was working in the lab, I was involved in lots of different projects with lots of different protein targets from the that were used for the structural characterization, for the drug discovery, for diagnostic devices, and as well for method validation. And we had as well collaboration not only with academia, but as well in industrial parts. So why do we need this protein? So why it's so needed and so interested isolated protein? So we can use it for the protein-protein interaction studies, for the antibody production, for the affinity specificity studies, for the DNA binding and activity assays, and as well what is our most interest is the structural studies. So how does it look like, the process? So this is as well one of the slides that Andrea as well previously showed you a little bit. So in the short, there are lots of steps to get the protein. So the first one is just construct design, that we have a lot of tools that help us to design the right constructs. Then we have to clone it, so it's molecular biology. And then we move to the delivery expression host, that could be the prokaryotes and eukaryotes, so bacteria, yeast, insects, mammalian. Then we have a step of finding the right small scale expression, so the right conditions for the expression. Then we are doing the scale up. Finally, we can purify the protein, and we have a purified protein. But this is not really the end of the process. You have to do a lot of quality control, because whatever downstream application you want to do, you have to be sure that all these processes and all the steps, you have the final product that actually you will be really happy with the results after, and you can be confident with your experiments later. So whatever we are doing in the lab, it's checking the purity. So the easiest is to see it basically visually on the gel. So you see if there are any contaminants, other bonds, if it's, degraded, or so if you have a pure protein, you can check as well stability. So, if it's fine in the solution that it is, if the buffers are fine. You are checking as well the homogeneity, the molecular weight. So we have some instruments that they allow us to do it. And as well, we will have soon another really nice instrument implemented thanks to PRP funding that will even speed up this let's say quality control step. We check as well for the molecular weight integrity, maybe for the post-translational modification with the mass. This is, we don't have in-house, we are outsourcing this part. And as well, we are checking if you are working on some enzymes or it has to be active protein, we check the activity. So after all those steps, then you can just apply your downstream application that normally in our laboratory we are interested in the protein-ligand interactions already, as well Sonia before, and as well Alberto was telling you all the application of the instruments. We will have as well implemented the microscale thermophoresis that we will have, that we can as well check the bindings, affinity, and as well then, of course, all the structural studies that as well Andrea was telling you, from X-ray crystallography to SACS and hopefully in future cryoEM. So this is just to give you an idea of actually success rate of just to get, for example, in the crystallography. So just to get the final class, final structure. So from all the design construct that you have, the one that you cloned, there are only 1/3 that they are getting expressed. Then half of it getting purified. Then to get the soluble protein, and then the one that it actually crystallized, it's really even lower. And that even if you have the crystal, it doesn't mean that they will diffract well. So then you will be able to have this final structure. So let's say the rate is quite low, but anyway, we are quite hopefully to, if you start a new project, to get where you want to get. So how do you start? So it is important to know your target. So you have to know if you are working on a prokaryotic or eukaryotic target proteins, what kind of quantities you would like to have, where is the native localization of this protein, so if it's intracellular, or if it's secreted, if it's membrane protein, what is the size of the protein? I mean, if it's a single domain or multi-domain, does it have any disulfide bonds, or it needs some post-translational modification, or if it needs cofactors that they will help with the correct folding or activity, or if it has any interaction partners, and as well, what is your application with the downstream? So this would help all this kind of question to help you to choose the expression system, let's say. So they are from the quite complex, like even animals, but let's say plants, mammalian cell lines, insect cells, and yeast, to very simple bacteria. So you can see that, I mean, so the speed and as well cost differs from the system as well. If you need to have the post-translation modification, of course, the simple system and the most cost-effective is the worst in this part. If it comes to the folding of the protein as well, the best are, of course, mammalian eukaryotic system and not prokaryotic. But actually, there is not universal expression system. So how do we start as well? I mean, if you find, I mean, you choose maybe the system that would you like to exploit. We are doing the construct design. So basically, this is an example of a protein. So it's quite big protein. So getting it a full length, it could be quite challenging. But with our case, we were interested of the part that is the kinase domain. So it's this active part. So what you are doing, you are analyzing, let's say, sequence. You want to know to create, I mean, if you would like to have this domain, so how long it should be or how to craft it. There are some tools that help you to predict if you don't have the structure, you don't know how does it look like really. There are some tools that help you to predict if there will be some helicases or beta strands. So then you are trying to choose, let's say, which would be the best domain to take for your studies. There are now, of course, with help with AlphaFold and with artificial intelligence, it helps us a lot in this step as well, because the prediction is really nice. But as well, like this is this protein exactly that we are seeing here. So you can see that it has really, I mean, this kind of N-terminal part is well, let's say, compact and well structured as well, C-terminal, but it has all this kind of really flexible loop. So if you would like to produce it, I mean, recombinantly, it's quite, let's say, difficult and challenging. But with how do you do it with AlphaFold? So basically, you can as well follow exactly the sequence of your protein, and you can see which is predicted to be this flexible, which you would like to maybe cut out from your construct to get a more promising protein expression. Other part you have to take in account in design is as well if you would like to fuse it with another protein. So we have as well this for the purification we are using these tags that they are called. So they are affinity tags that we have as well some resin. So we just thanks to them we can choose those from all of the host other proteins. We can capture them and help them with the purification. So of course here is the whole game of choosing actually which would be the best tag. So it depends maybe what you have in the lab, if it should be on the C-terminal part of your protein, on the N-terminal, if you would like to have maybe a double tag, if you would like to have a cleavable tag, or maybe you would like to use it for the downstream application. So use it as an anchor if you would like to have some studies with the interaction. And some of them, especially in the prokaryotic system, they help you to influence the expression and help with the solubility and as well the purification yields. So if you are already ready with your construct, you know how you would like to have it, the molecular biology starts. So basically your gene of interest has to be transferred in the vector, and then later on you are using in the certain expression system. So we here in the laboratory, we have almost 60 expression vectors. So they are as well different cloning methods. So yeah, I mean, the library has the vectors with the different tags. So there is a lot to choose from. As well, we have lots of library of expression vectors for the insect cells and as well for the mammalian cells. So as well, you have to take in account if you would like to have, if you have a single protein or if you would like to express a complex. So you can do it as well in the single vectors. But what we have, especially for the baculovirus or in insect cell expression, so in the eukaryotic system and mammalian system, we have this kind of nice set of vectors that they allow us to actually create this kind of multi-vector. So you have you can express at the same time on the same vector partners for the complex. So it can accommodate a lot of different inserts. So I was telling you about this insect cells expression system. So this is the one that actually we implemented it in the laboratory. So it is based on the viral infection of insect cells that they are from the family of Lepidoptera. The most common virus is used, Autographa californica, and it is double-stranded DNA, supercoiled, that it's quite big, and it has the capability of actually accommodating quite large insertion of DNA. So it is, we take advantage of exchanging the polyhedrine that it's expressed really strongly with our gene of interest. So this is basically the overview. In a laboratory, we have two different, let's say, techniques of getting your gene of interest to the baculo DNA. So it can be from the homologous recombination, so it occurs in the insect cells itself, or we create this backmeat with our gene of interest in E. coli already before, and then we do the transfection of this backmeat to the insect cells. So this is just to give you an overview of the timeline and the workflow of those two systems. Anyway, I mean, there are certain steps that they are a little bit different, but anyway, you can see that it's quite long process. So to get the pellet, let's say, the scale-up pellet to do the protein purification, we need at least 20 days. So just to give you an idea, for simple systems like E. coli, you need the same, I mean, you are at the same step after 1 week. So what do we do next? We do the expression test. So as well, in our laboratory, we have different cell strains of E. coli. We are testing at different conditions from medium temperatures, inducer concentrations, as well time points. With the insect cells, we have 3 different cell lines as well. There we are checking the temperatures, time points, and volumes of the virus. Mammalian cells, we for now have the one cell line that is in suspension, but in the future we would like to expand as well this field. So normally we are doing it in the small format, so it's 24 well plate. So just to give you an idea, exactly, I mean, if you have this one construct, you just decide on this one. If you would like to check, I don't know, 4 different tags, then different cell lines and mediums to different temperatures, you have already ending up with 32 samples just to test out from the one construct. So it's quite difficult to handle it manually. So what we are having, we have this kind of liquid handler, so it helps us with automatized protein purification from the expression. And then finally, if we find our conditions, we have this kind of scale-up possibility in our laboratory. So for E. coli, we have 2 incubators that each can accommodate from 10 to 12 liters. And as well, we have the fermenters that can accommodate 10 liters. The similar thing is for the insect cells. And now with the PRP project, we will have 2 additional very robust incubators. And for the mammalian cells, we can do 1 to 2 liters. So then afterwards, when you have your expressed protein, you have to purify it. So you have to get out from the cells of the host that you would like, that you expressed it. So as well in the laboratory, we have the instruments for the lysis. So we have the orsonicator of homogenizer. Then depending exactly on your constructs, or depending what kind of tag you have, affinity tag, we have a set of different resins. Then as well, depending on your construct, we are normally cleave out the tag. So whatever it's not really coming to your native protein, for the structural studies, you would like to have as simple protein as you can get. And then we have all the other techniques as well to polish a little bit the purity. And finally, as well, size exclusion chromatography that gives you idea as well about the colorimetric state of your protein. So for this one, we are using the instruments, FPLC. So as well with PRP project, we will have, fundings, we will get 2 more instruments. So it will give us a lot of help as well with this one. So this is just as well the summary of how many steps there are inside of this pipeline. So from the strategy design, so what is important to take in account, or for the cloning, for the small scale, to finally to have for the downstream applications. So I will give you a little bit of examples how, for the proteins that we were expressing in our laboratory. So one of it is ULK. So already Paola was telling a little bit something about it. So it's crucial key player in the initiation complex of autophagy. So it was linked, so inhibiting this protein, it was linked with a good outcome for the anti-cancer therapy development. So we had, let's say, so this is exactly the whole full-length protein. So we were interested in the kinase domain, as well as the C-terminal domain, that it is the part when all the other interactors of the complex bind. So it was done. So after the analysis of, prediction of the secondary structure, we were creating a lot of, previously in the lab, there were a lot of constructs created. So for the N-terminal part and C-terminal part, so with then as well cloned with the different vectors and different cell strains were checked. So you see it's a lot of work, and in the end, none of it gave a good, let's say, good outcome. So meanwhile, the crystal structure of this protein came out. So we understood from the literature that actually the most so what the trick was to get the soluble and stable protein, it was actually expression it with the lambda phosphatase, because this is a kinase, so it was active while expressing in the E. coli, and it was toxic for the host cells. So expressing it with the lambda phosphatase, it gives you the possibility to get a nice amount of good quality protein. So we use it for a library screen of the different inhibitors. We chose some of the inhibitors that they were very promising, and we wanted to crystallize it. But unfortunately, this wild type protein is quite not so prone for crystallization. We were able to obtain only one crystal, and they were not really diffracting quite well. So what we decided is just to understand how we could optimize the crystallization. So what we could do with the protein to get the better crystals. So we had the wild type that it has good protein production yield, but the poor diffraction crystals. So from the first, let's say, structure that was published, so from the paper, they did the surface entropy reduction part that they wanted to see maybe if there is any mutation that they could introduce to favor the crystallization contact. So they chose those mutations, so they were able to get good crystals with the diffracting, but unfortunately the protein production was very, very low. And in our laboratory, we were actually struggling to get enough material to do the structural studies. So what we did as well on the already existing then the structure. We did this the analysis of the surface. So we were seeing here that this is the active site, so far away from the active site, but could influence the crystal contacts. There are those residues. So we decided to mutate them to something simpler, to the alanine. And actually, it gave us quite good results. So we were able to obtain good yield of the protein. We obtained quite well diffracting crystals, and it was quite prone to crystallization. So we were able to obtain it with the different compounds. So another example is this protein targeting to glycogen, that it's linked to the Lafora disease, that it's fatal neurodegenerative disease that is caused by accumulation of abnormal insoluble glycogen, that they are called Lafora bodies. And for now, there is no cure. So, it was shown that actually this PTG is one of those proteins that they are subunits for PP1. As PP1 is, protein phosphatase 1 is present in the loads of different cellular processes. And its specificity it gets from this kind of regulatory subunits that they are called. So this one, and then like a PTG, it brings it to the right substrates, let's say. So PTG brings the PP1 for all the substrate with the glycogen metabolism proteins. So it was seen that the metabolism of glycogen, in fact, in this Lafora disease is altered. And it was shown in the studies that knocking out this PTG protein, it gives almost complete disappearance of Lafora bodies and the resolution of the neurodegenerative disease. So it would be quite interesting as a target. So what we were interested in to know if the destruction of this complex could help in development of the therapy. So we knew that there is a structure of PP1, but there was no structural information about the PTG. So we decided to try to express it and to get it. So this was the standard experimental strategy. So we had, of course, we were mostly interested in the full-length protein, or the protein that has as well the part that actually it is responsible for the interaction with the PP1. So when we were trying to express this with lots of as well efforts, it was not really good, giving us the good results, as it has all this kind of N-terminal flexible part. So it's normally not very promising as a target for the expression. So unfortunately, we had a really low yield, and we were thinking, okay, it would be not really enough to do all the studies that we wanted to do. But we ended up as well trying another trick. So it was the co-expression with the chaperones, and we obtained enough of the soluble protein to actually get to be able to do all the downstreams that we were interested in. So actually, the yield went up on tenfold and allowed us to get the good experiments. And from the same project, we were as well working on the not getting only a separate proteins, but as well, I mean, working on the complex. So we were interested in obtaining the complex. So the strategies were that one protein has a different different tags, as well different antibiotic resistance. So here you can check, I mean, if this is a really simple test as well to understand if this is really interacting, those proteins are interacting. You this kind of, you can see it on the gel. So as well, with the two different resins, so one would be recognizing this GST, another the HISTAC. So when you are loading on one or another, anyway, even though one, it shouldn't bind to this resin, you always have the complex. So we confirm that we are able to purify this one. So after a lot of as well work on getting the right let's say amount and lots of crystallization trials, we were able to obtain finally the structure. And what was as well nice, it is this comparison with like experimental. So our is this kind of the yellow part with how was it predicted by AlphaFold. So the part that was really well structured, they predicted really, really well. But this N-terminal, that it's exactly quite flexible and it is responsible to create a complex. It has some issues. So it was predicting some alpha helix. In fact, and we had here some beta strands. So this could be as well, I mean, experimental part. So you can use it for a more training of the system. So the last example, what I wanted to show you is the protein complex production in the insect cells. So we had this nice collaboration recently that our collaborators are working on PROTAC degraders. So it's quite interesting technology. So it's composed of, let's say, two parts. So you have one part of this system that recognizes E3 ligase, and another one is your protein of interest, and it brings your proteins of interest to the proteasome. So it makes the proteolyze. So our collaborator had already the target protein, and it had this kind of, and it had the inhibitors for, or the molecule that binds to their target and the molecule that binds to the E3 ligase, and we needed to produce the E3 ligase to get the complex. So we've seen that already the system is, I mean, it's validated. So we were able to see from the literature that it was expressed in the insect cells. There is this really nice as well bank with the vector, so we could actually get the vectors from this bank from the publication that they set up all the system, and we followed the protocol that was published. So they were expressing it those two proteins separately, but actually in our hand we had the problems with obtaining one. So we understood that actually it is expressed, it is soluble, but it doesn't bind to the resin. So we tried different resins. We tried as well the co-purification, because sometimes if it's a strong complex, it could be favorable to do at least co-purification, and unfortunately not even this part helped. But co-expression, so just do in the same moment the same proteins together, it was finally gave us a good results, and we were able to obtain a very good quality complex. So now I can follow with the crystallization part. So this is all the collaboration, not all the collaboration that we did with this within this project. So thank you very much for your attention.